Yes, I'm ready now. Can you see my slides?
Yes.
Okay, so let me start my talk. My name is Takahiro, and I work as a research associate in the QuizTech lab at McGill University. In this talk, I'd like to present the result of our research on genome-wide identification of single nucleotide mutations from time series mutation accumulation data. Single nucleotide mutations are the finest mutations important in evolutionary studies. The rapidly improving high-throughput sequencing technologies provide unprecedented opportunity to identify single nucleotide mutation across the genome. However, it is difficult to confidently identify single nucleotide mutations from high-throughput sequencing data due to high sequencing error rates, mismapping of sequencing lists, and variable depths of coverage. Different researchers take different approaches, and the performance of various approaches is not well understood. In this study, we developed a method called Bayesian mutation finder, which is available at the following GitHub link. The method is designed for accurate genotype calling at single nucleotide site for mutation accumulation MA data with moderate depth of coverage. It calls genotypes using BGC and filters genotype calls using a binomial test, where we examine the goodness of fit between observed nucleotide read counts and their binomial expectation in each genotype call. To assess the performance of the BMF method, we compare the performance of the method with that by GATK using time series mutation accumulation and simulated data. For this, we employ two matrices, false discovery rate and positive predictive value. With empirical data, we took proxies of these two matrices, which are cross-line and within line sharing of candidate mutation. The idea here is that true mutations are expected to be shared between time points exclusively within ML lines. On the other hand, with simulated data, we took exact estimates of these two matrices. This table shows the time series mutation accumulation data we analyzed. We have 3 time points, and we analyzed 14 Daphne-Pirex ML lines with data at 3 time points. Here we show mean generations, read lengths in base pair, total coverage, which is sum of depths of coverage over the ML lines. Here we show the minimum and the maximum total coverage at analyzed sites within square brackets. This way, we avoid analyzing sites with total coverage deviating from typical values. The last column shows mean coverage. Here we find that the depth of coverage in our data is moderate. To process the data we analyze, we trimmed adapter sequences using Trimomatic. We mapped sequencing leads to the Daphne-Pirex reference sequence PA42 version 4.2, concatenated with the Daphne-Pirex mitochondrial DNA sequence using BWMM. We kept only properly paired primary leads with at least 20 mapping quality scores using some tools. We removed duplicate leads using some tools and GATK. We excluded leads that mapped to mitochondrial DNA using some tools. Furthermore, we removed these potentially problematic sites. We removed tri- and tetra-allelic sites, site with total coverage deviating from typical values, site with error rate estimate greater than 0.01, in BMF only, because error rate estimates are not available in GATK. Sites involved in putative repeated regions identified by repeat masker. This figure shows the comparison of cross-run sharing of candidate mutation by BMF and GATK. Here, the result level GATK show GATK result with generally recommended parameter cutoff values in hard filtering. The result level GATK adjusted show GATK result with adjusted parameter cutoff values in hard filtering. The y-axis shows the rate of cross-run sharing of candidate mutation, which are proxies of false discovery rates. By looking at this figure, we find that BMF had no cross-line sharing of candidate mutation, indicating that BMF may have low false discovery rate. On the other hand, GATK had some cross-line sharing of candidate mutation. Although adjusting the parameter cutoff values in hard filtering somewhat decreased the cross-line sharing of candidate mutation, the GATK method had much higher cross-line sharing of candidate mutation compared to BMF. This figure shows the comparison of within-line sharing of candidate mutation by BMF and GATK. Here, y-axis denotes rates of within-line sharing of candidate mutation, which are proxies of positive predictive values. Here, we find that BMF method has much higher rates of within-line sharing of candidate mutation compared to GATK, especially when GATK method uses generally recommended parameter cutoff values. Although adjusting the parameter cutoff values in hard filtering somewhat increased the rate of within-line sharing of candidate mutation, they remain much lower compared to those by BMF. Using the candidate mutation identified by BMF, we estimated mutation rate at each of the 3 time points. Here, the unit of the mutation rate is per site per generation per MLI. In this table, standard error of the mean is shown within parentheses. The mean of the mutation rate estimates over the time points, which is 4 times 1 times 10 to the minus 9, agrees well with the previous estimate by Keith et al., which was 3.8 times 10 to the minus 9. Based on high coverage, mean 36x sequencing data. This figure shows the comparison of computational efficiency by BMF and GATK for analyzing ML data at time point 1 under the same computing environment. Here, we found that BMF was 131 and 464 times more efficient than GATK in terms of computing time and memory consumption, respectively. To assess the performance of the method when the 2 mutations are known, we generated simulated data and applied both methods to the same data. For this, we use NEAT and the Daphne-Plex reference sequence, PA42 version 4.2, which was used in the empirical data analysis. Here, we generated data of independently evolving 14 diploid individuals. Paired and 150 base pair sequencing leads with the mean 10x depth of coverage was specified to mimic the empirical data we analyzed. This figure shows the result of comparison of false discovery rate by BMF and GATK method. Here, the false discovery rate was calculated as the fraction of candidate mutation that were not true. GATK result used generally recommended parameter cutoff values in hard filtering. Consistent with the result with empirical data, we found that BMF had lower false discovery rate than GATK. This figure shows the result of comparison of positive predictive value by BMF and GATK. Here, the positive predicted value was calculated as a fraction of candidate mutation that were true. Also consistent with empirical data result, we find that BMF had higher positive predicted value than GATK. Although we know that here with simulated data, both methods had much higher positive predicted value compared to the result with empirical data. These are the conclusions in this talk. Performance comparison under the complexity of empirical data was enabled by time series mutation accumulation data. Lower false discovery rate and higher positive predictive value by BMF than GATK. Fewer parameters requiring adjustments specific to analyze data by BMF than GATK. Much faster computation consuming much smaller memory by BMF than GATK. Much lower positive predicted values with empirical data than with simulated data by both methods due to moderate coverage and mismapping resulting from existence of many large duplications. Thank you for your attention, and I'm happy to take questions.
Excellent, very nice talk. At this time, if you have a question for our speaker, you are welcome to use the raise hand function to raise your hand to be able to ask that question verbally. If you would prefer, you are also welcome to type your question in the chat where we will read it out and have our speaker answer it verbally. I was a little curious about kind of the use of the term positive predictive values. Can you say a little bit more about what that means and how that is measured?
Yes, so positive predictive value measures the rate of candidate mutation that were true. So with empirical data, we calculate the fraction of confirmed candidate mutation at later time point among all candidate mutation, because we don't know the true mutation in empirical data. But on the other hand, with simulated data, because we know true mutation, we calculate the positive predicted value as fraction of candidate mutations that were true. So this basically measures the power of the method.
Okay. Excellent. Okay, that makes sense. That makes sense.
Sir, is this BMF package, available as a package, R package, Python package?
Sorry, could you say it again?
Is this available as R package or Python package?
So this is basically written in C++.
C++, okay.
Yes, so yes, I have this GitHub link.
Okay.
And I have instruction on the website.
Okay, interactive website.
Excellent. So it's a standalone application then?
Yes.
Excellent.
Yes, we also use other packages, but yeah, the instruction is available at this GitHub link.
Excellent. All right, I think we have to move on to our next speaker. Thank you very much for your talk and for answering all of our questions. Jacopo, if you would like to get your slides shared, I will let you know when to start talking.
Yes. Okay.
Excellent. Those look great, and you can start whenever you're ready.
Okay. So, hi everyone. Thanks for joining this talk. I'm Jacopo Martellossi. I'm a PhD student at the University of Bologna. And today I will talk about the contribution of transposable elements in the emergence of within and between individual structural variants in bivalve mollusks. So first of all, why should we study structural variants and transposons? Well, because there are quite a lot of reasons about this, but in recent years there are increasing number of evidences showing us how structural variants and transposons can impact on the biology of organisms. For example, they can promote speciation, they can change the chromatin landscape of the genomes, they can promote adaptations to local adaptations to novel environments or stressor, and they can trigger, transposons can trigger large and complex structural variants thanks to the creation of homologous regions across the genome. So my case study are bivalves and mainly oysters. Why bivalves and oysters? Because bivalves are there are a lot of bivalves that are economically important species. They are worldwide distributed and they possess a highly diversified TA content. Oysters in particular, for oysters are the most well-characterized bivalve species because of their economical importance. We have 5 high-quality chromosome-scale genome assemblies produced using different long-read sequencing technology on ONT, CCS, and CLR reads. And moreover, for 2 of these species, we also have resequencing data coming in luminary sequencing data. So our first question was to try to understand at what extent, or better, how much structural variability we have within individuals in oyster genomes and how this variability arises. So what do we mean with within individual variability? We just mean heterozygous structural variants. Just like SNPs, every time that the structural variants arise, it will be present in a heterozygous state. For example, for transpositions and deletions, or insertion, transposition and deletions, when transposons move to a new location of the genome, it will randomly jump in one of the 2 homologous chromosomes. So it will create an heterozygous insertions, and this insertion can also be seen as an hemizygous genomic region. So basically a genomic region that is present in only one of the 2 homologous chromosomes. So to try to answer to this question, basically we have remapped long reads used to assemble the genomes against the genome itself using 2 different mappers. So here is important to note that the assemblies are haploid representations of diploid genomes. So we assembled only one of the 2 homologous chromosomes. Then we have called within individual structural variants using 2 different SVS scholars, 2 popular SVS scholars. So we have 4 different combinations of mappers and SVS scholar. Then we have benchmarked our SOS calling pipeline using simulations. And finally, we classified within individual structural variants based on type and amount of overlap with transposon annotation. Okay, sorry. So here in the panel A, you can see various kind of heterozygosity estimations for 4 out of the 5 species analyzed. The 5 species is still ongoing. I'm sorry for that. But anyway, for bivalves are quite known to be highly heterozygous species. This also put some problems in genome assemblies. And as we can see, we don't have only high SNP-based heterozygosity, but also high level of structural heterozygosity that can reach up over 13% of the Crassostariacensis genome, only keeping into consideration insertion and deletions. So basically, for example, for Crassostariacensis, we can say that the 13% of its genome is present in an hemizygous state. So it's present as single copy in a diploid genome. Here, there is just an high-view snapshot about one of these regions in which there are three clear hemizygous deletions, so heterozygous structural variants that show half of the coverage compared to the surrounding genomic regions and are depleted by SNPs. When we look at the overlap between transposable elements and hemizygous deletions, here we deeply characterize deletions only because we obviously have the gene and transposable element annotations for the reference chromosome, so we can well characterize these genomic regions. These genomic regions are well characterized, and as we can see, we have very high D content across these genomics regions that can reach up to the 100% median and more than the 50% in mean. And as we may expect from highly diversified genomes in terms of TE content, when we look at the composition of these deletions, we can see that they are composed by almost all kinds of different TE types, so from DNA up to science, with a higher percentage of DNA unrolling cycle for Crassostrea compared to Ostrea, in which we have more retrotransposons. And this is in line with their gene-wide distribution of transposable elements in the two genera. Because of this very high estimations, we decided to benchmark our pipeline using simulations and different reciprocal overlaps between our true set and our cold set. So here in the panel A, we have the precision rate at these different reciprocal overlap requirements and for different kind of reads. So CCS, ONT, and CLR. On the B panel, we have the recalling rate. So as we can see, our merge set, which is this last column here, as we may expect, have higher precision rate compared to all other combination of S2S callers and logarithm mapper. Whereas for the recalling rate, we, especially for CLR and ONT reads, we have really lower recalling rates. So basically, you have a lot of false negatives. However, it's interesting to see that for PBSV, which is an S2S caller, we have very high recalling rate compared to sniffles at very high reciprocal overlap requirements. So this means that PBSV is very good in the correct estimation of breakpoints of variants, which is breakpoint estimation is the most difficult part in structural variance calling. So then we wanted to classify these TE-related heterozygous deletions based on the type and the amount of overlap with transposable elements. So here you can see the different types, 4 different types. Okay. Yeah. And when we look at the number of elements that fall within each category, we can see that in Crassostrea genus, we have pretty high, we have pretty high, we have pretty high number of putative T insertions, and whereas for Ostrea, we have an equal amount of putative T insertions and internal T deletions. And when we look at the divergence of, at the age of the transposons that are present within these regions, we can see that we have very younger transposons within putative T insertions and full T deletions compared to between T deletions and internal T deletions. This means that these classes here likely contain novel T insertion, whereas between T deletions and internal T deletions, more frequently occur within older transposons. So here the take-home message is that both putativity insertions and deletions contribute to within individual differences in bivalve mollusks. So as the second question, I will go very quick here. We wanted to understand if structural variants also contribute to between individual differences in oysters. For this, we have chosen Crassostra-Gigas as model system, for which we have quite a lot of free sequencing data coming from 3 different locations in China, from Korea, Northern China, and Southern China. We have applied a quite stringent SOS calling pipeline using short reads data. And then we have inferred population structure using both SNPs and structural variants. We have identified T-related deletions, and we have tried to identify deletions under putative selections. Why we characterized only deletions here? Because for short reads technology, insertion cannot be fully resolved usually, so we cannot have the representation of the insertion in terms of nucleotides. And moreover, they are also more prone to false positives as well, and the same for inversions and translocations. For these reasons, we decided to only rely on deletions. So here we can see the results of the two analyses using deletions only and SNPs. So here we have admixture proportion using with 2 ancestry group, and here we have PCA analysis based on deletions and SNPs. And as we can see, they very well reassembled each other. So we have always samples coming from the south of China that are well differentiated from samples that come from the north of China and here from Korea. And we have the same situation here for SNPs. So here the take-home message is that SNPs, we can use deletions to infer population structure, and we have, as we can do it with SNPs, and we have deletions that are differentiated, that differentiate populations. So when we characterized these deletions, we found that the 46% of population informative variants are T-related, so they have a minimum of 70% overlap with the transposons. The median T coverage of population informative variants is equal to the 60%. And moreover, we found that 1,424 candidate deletions under putative selection using very simple FST outlier analysis. So these are preliminary results, but anyway, we have found some evidences of selection acting on structural variants. So time is running up, so I will not recap all the take-home message. I just would like to thank you for the attention, and also to thank my PI, Fabrizio Ghiselli, Alexander Su, and Valentina Peona that follow me during my abroad period at Uppsala University, and my collaborator, Andrea Lucchetti, as well as all the Evocom lab at the University of Bologna. Thank you.
Right, excellent talk. So sorry to have given you a premature.
Okay, don't worry. I was
yeah,
afraid
no,
already.
Entirely my fault. It's been a long couple of days. If anyone has questions for our speaker, now would be the time to raise your hand and ask those questions. And while we're doing that, our next speaker, I believe Mariangela can go ahead and put up her slides. Seems everyone's feeling a little bit shy at this late hour. Thank you so much for your talk. And Mariangela, I'll let you know in just a second when we are ready to have you get started. All right, you're welcome to start whenever you are comfortable doing so.
Perfect. Thank you very much. And first of all, I would like to thank the organizers for this virtual conference. So my name is Mariangela Iannello. I'm a postdoc at the University of Bologna, and today I'm going to talk about longevity. So what determines how long we live? This is a very simple question, but of course the answer is very complex. And that's because the mechanism of aging is very complex and poorly understood. It depends on genetic and environmental factors. Anyway, it's believed that the primary hallmark of aging is the accumulation of damages in cells. So an increased number of mutations on DNA, errors in replications and transcription, accumulation of misfolded proteins in the cell. Anyway, organisms have evolved a different mechanism to try to limit the accumulation of such damages. So, for example, we have a mechanism of DNA repair, different responses to stress, pathways involved in the stabilization of protein folding. Anyway, when these mechanisms are not sufficient to contrast the accumulation of damages, we have dysfunctional cells. And this leads to programmed cell death or apoptosis, and eventually this leads to the death of organism. Anyway, we know that some species live longer than others. And here you can see some example of extreme longevity in different animals. Now, some animals are very good models for studying aging and longevity, while other animals have never been considered for studying longevity. And considering that longevity is very complex, is multifactorial, and of course poorly understood, it's important to consider additional species for studying longevity and aging. And a good example can be bivalves. For example, bivalves include species with extreme longevity. We have, for example, Arcticus landica, which is the longest lived non-colonial metazoan known so far, and it can live more than 500 years. And also bivalves are very interesting because the Bivalvia class shows the highest lifespan disparity within an animal class. Anyway, in this context, bivalves have been overlooked. And for example, there are no works about molecular evolution of genes in bivalves with different lifespan. And so that's what we decided to do. So we investigated molecular evolution in bivalves with different lifespan to try to infer genes that may be involved in the extended longevity phenotype. And here you can see the species that we investigated. In red, we have long-lived species. So besides Arcticus landica, we have a few other species that can live more than 150 years. While all the other species usually live from a few years to a few decades. And just briefly, here what we did, we analyzed transcriptomes because unfortunately we do not have a lot of sequenced genomes for this species. And we inferred open reading frame, we performed an orthogroup inference, we analyzed one-to-one orthogroups, and at the end of the pipeline, we ended up with 2,200 orthogroups. Now, I don't think I have much time to go into the details. Anyway, if you are curious about the pipeline, just let me know. So the aim of this work was to investigate convergent molecular evolution in long-lived species to try to analyze genes that may have a role in the extended longevity phenotype.
So we investigated convergent molecular evolution at 3 different levels, which we think are complementary. First, we investigated convergent dn over ds.
This is a branch test. So it means that we are analyzing different selective pressures acting on the whole sequence. And the aim of this analysis
was to investigate genes where dn over ds changed convergently in long-lived species compared to the short-lived species. And then we analyze also convergent positive selection. So this time it's a branch site test. So it means that we are investigating signature of positive selection at sites. And the aim of this analysis was
analyze genes with positive selection in long-lived species. And finally, we investigated convergent amino acid substitution. So this time we are shifting the focus to convergent phenotypes. And the aim of this analysis was to identify genes in which substitutions in long-lived species have independently occurred toward the same or similar amino acids. And we want to make sure that the convergent signal we were investigating was specific of long-lived species. So we ran the same analysis 10 additional times, but this time setting as foreground short-lived species instead than long-lived species. So anytime a gene was significant in the long-lived species and in any of the control datasets, we removed that gene from the next analysis. So once we got our genes with a significant convergent evolution in long-lived species, we checked if those genes had been previously associated with longevity in other animals. So we investigated a couple of longevity databases, and also we analyzed if these genes were involved in pathways that may have a role in longevity. So, for example, quality control of DNA, RNA, and proteins, response to damages, apoptosis, and so on. So here a couple of results. So we found 277 genes with convergent DN/DS, and you can see that most of these genes show convergent decrease in DN/DS in long-lived species. So it means that DN/DS in long-lived species was lower compared to short-lived species. And so it seems that these genes are subjected to a stronger purifying selection. And then we found 33 genes with convergent positive selection and 27 genes with convergent amino acid substitutions. And to better characterize these genes, we performed a functional annotation and an enrichment of geoterms, and we found some interesting enriched geoterms. So, for example, involved in apoptosis, response to damages, response to oxidative stress, protein degradation, and so on. So briefly, I will show you some interesting genes. For example, we found some genes with convergent DN/DS, which are transcription factors involved in the control of cell proliferation. So these genes are constrained by a stronger purifying selection. And these genes in other works have been associated to tumor progression. So mutation in these genes or an abnormal transcription has been associated with tumor progression. So now tumors are common in bivalves, but they are rare in long-lived species. So it's interesting to see this convergent evolution in these genes that have a role in the control of cell proliferation in bivalves, in long-lived bivalves. And also we found some genes involved in protein degradation, both with convergent positive selection and convergent amino acid substitution. And now we know that the accumulation of damaged protein is an hallmark of aging. Maybe this signal could be involved in a more efficient handling of protein degradation in cells. And also we have some gene involved in DNA repair, and in other work so far, in other taxonomic group, positive selection was found in genes involved in DNA repair. So this could be a common feature in different species, different long-lived species. And finally, I would like just to highlight this gene here, HIF1, which is a transcription factors, which responds to low amount of oxygen or increased reactive oxygen species. And it's considered a master regulator of longevity because an increased transcription of these genes usually leads to extended longevity in some other species. So this could have a role in longevity as well in bivalves. Anyway, looking at these single genes may be interesting. Anyway, as we said, longevity is complex and multifactorial, so it's unlikely that a single gene may be responsible for the extended longevity phenotype. And instead, it's more likely that a combination of different genes and different pathways may have a role in this phenotype. And for this reason, we investigated the network of protein-protein interactions among the whole genes showing convergent
in long-lived bivalves. And here, this is the network we analyzed. And the first thing we saw is that the number of interactions in this network is statistically higher than expected. So this means that these genes are biologically connected. And also, it's interesting because we found these genes using different approaches. So a combination at different methods, particularly in this case, if we wanted to investigate convergent evolution, so using different approaches may be very effective, particularly when we want to investigate complex phenotypes. And in bold, you can see genes that have been previously associated with longevity in other animals. And we found that the number of genes previously associated with longevity in this network was higher than expected. So what does it mean? It means that these genes may have a role in longevity also in bivalves, but more importantly, that the mechanisms beneath extended longevity may be similar across metazoans. And so what about all the other genes? So the other genes that have never been associated with longevity so far. Is it possible, but they are in the same network? So is it possible that these genes have a role in longevity in multiple species as well, or they are specific? They have a specific role in extending longevity, maybe in bivalves. So hopefully we will investigate
better in the future. Okay, just to conclude, I would like to thank all the co-authors, all the guys of the Evolutionary and Comparative Biology group, and of course, thank you all for the attention.
All right, excellent talk, and wonderful to be able to put the two of you right next to each other. If anyone has questions at this time, you are invited to raise your hand so that I can invite you to unmute. If you're feeling a little bit shy, you are also welcome to put questions in the chat for us to read off and have answered by our speaker. All right, so we have a question in the chat, and that question is, how much overlap did you find between the different methods to identify the genes?
That's a great question. I didn't mention the overlap, basically because I don't think these methods actually overlap a lot, because, for example, you can have a convergent DN/DS in one gene, maybe toward purifying selection, but still this gene may have a signature of positive selection, but you can have also the opposite, so not overlapping at all. We do not have a lot of overlap. So it happens that some gene show signature of positive selection and also convergent DN/DS, but it's not a common common situation. So few genes overlap.
Perfect. My son is working on some jewelry making right beside me, so that was that sigh you heard. And another question from the chat, are you planning to investigate the same signature in other groups?
I hope so, yes, that's the plan. We would like to investigate the same, to do the same analysis also in other metazoans, yes.
Wonderful, thank you so much for taking those two
Thank you very much. Bye.
Yeah, so we're gonna go ahead and start our changeover to our next speaker. Philip, if you would like to share your slides.
Yes, of course. Okay, so screen sharing is still activated.
It should be for you, yes.
Yes, 1 second.
Mhm.
Okay, here we are. Okay, I hope you
Wonderful.
can see me
Yep.
and hear
You
me.
are welcome to start whenever you would like.
Okay, so thank you. Then I will start right away. Thanks for attending my talk. My name is Philipp Resel. I'm a postdoctoral researcher at the University of Graz in Austria. And today I will be talking about a small software project that we've been working on with the aim to facilitate a large-scale phylogenomic analysis. And we call this thing Velociraptor. But let's first zoom out a bit and recapitulate quickly what like a typical phylogenetic analysis may look like. So one of the first steps would be to gather some sequence data, which would be like from public databases or from own experiments. Then you would combine those into a file and then align those files. Afterwards, typically you would remove ambiguously aligned sites from that alignment and then calculate a substitution model that best fits the data. And after that, you would calculate a phylogenetic tree. So, and if we work with full genomes or transcriptomes, then we would have to add one additional step at least, which is a step to identify loci in the genome and establish orthology relationships between different sequences from different genomes. And all the other steps can necessarily be, theoretically be the same. So, but despite all these very clear steps really, and they have been researched for quite a bit, and there's lots of software available for all of these things. When you look at the literature, you see that analysis typically have very specifically tailored workflows. We were wondering why this might be, and I think there's some reasons, possible reasons to that. One thing I already mentioned is that there are many software packages available, and you may have your go-to aligner that you always want to use, or your go-to tree reconstruction program. And of course, you have a full range of different parameters to change the behavior of software. So you may be familiar with one set of parameters, but not so much with other sets. Then, of course, analysis like this depend on the computational infrastructure that is available. So, of course, rerunning things and testing different softwares can sometimes be quite challenging if you don't have access to large high performance clusters, for example. And also, as the amount of data grows, it can be increasingly difficult to organize this data. But what the result of all of this is that analysis that are produced with very specifically tailored workflows, relying on personally written scripts and so on, they kind of end up with reduced reproducibility and reusability. So the reusability is one thing. Imagine going back a few months after you perform some large-scale analysis, it might be quite difficult even for you to find all the different scripts and files and so on to rerun this. And if you move to a different lab, then the computer may be different and everything may fall apart in a way. And reproducibility, of course, is a big challenge in all of science. And of course, we want to make our analysis reproducible as well. So this is why we wanted to address some of these issues with our pipeline, which we call Velociraptor, the Rapid Phylogenomic Tree Calculator. And what you see here is the greeting screen, so to say, when you execute the command for the first time. And so there are all kinds of steps involved in a Velociraptor analysis, and I will go into detail a bit more in a bit. But first, I want to talk a bit more about the motivation behind our software and the design goals. So first of all, we aim for minimal software requirements. So Velociraptor only has two dependencies. One is SnakeMake, and the other is a container runtime environment, Singularity, or you can also use Docker if you want, because installing and compiling and maintaining software can be notoriously difficult. And we wanted to take this burden off the user who wants to focus on the science and who wants to play around with the data more than just spending time installing software. Another thing is we aim for maximum reusability and reproducibility and also platform independence. So our system, our software is operating system agnostic. You can run it on Linux, on Windows, and also on Mac OS if you want. You can interact with different HPC job management systems, and also sharing the data is quite simple. You just have to transfer the folder where the analysis was performed to another computer, and then you can basically rerun the whole thing again. And so the overall goal of this is we wanted to make phylogenomic analysis more explorative than they currently are. It should be easy to test different software and parameter combinations, and results from analysis should be easily compared. And so I think we should start to move towards treating computers as labs. And when we run programs, we perform experiments, and we have to repeat these experiments and test different, modify these experiments to see how robust they are. So after that, we can come back to a typical Velociraptor analysis, and you can see these steps that I mentioned in the beginning here already. They are mirrored in Velociraptor as well, with these different by running these different subcommands. And I will walk you through this very quickly now, what all the steps are doing. So first of all, we need to gather some sequence data, and sequence data in the Velociraptor sense are either genomes, so genome assemblies, transcriptome assemblies, or sets of proteins. You can use the NCBI genome browser, for example, browse your favorite organism group and download a CSV file, and you can almost directly input this into Velociraptor. You just have to add one additional column. You can specify specific assembly accessions. So to be very precise about the version of the assembly that you are using, we have some sanity checks for taxon ID correctness and species names and so on by querying the NCBI taxonomy database. And it's also easy, of course, to incorporate locally provided data. So, and when you run a command like this, all of what I've just mentioned will be done for you automatically. After this is done, so once we set up the analysis, we can start to identify loci and establish orthology. We do this by using BOOSCO at the moment, but we want to expand this also with other orthology inference methods in the future. So orthologous genes will be searched in each genomes, and then the completeness is assessed. And with this way, we can already filter out genes and taxa if you want, and all the data, it will be combined into separate files that can later be aligned. As I just said, so alignment would be the next step. So we would take these single copy ortholog files from different species and align them. And then we can remove ambiguously aligned positions with trimming. And we provide many different aligner and trimmer combinations. So you could use MAFT, Close-Tallow, or Marcel, for example, for alignment, and also 3 different approaches to perform alignment trimming. And in the process of all of that, we calculate statistics of the individual alignments, including parsimony informative sites and variable sites and so on. So you can then look at those statistics and go back and perform another set of trimming if you want to remove some additional alignments or some additional alignment positions. So once we are happy with our alignments, we can perform a substitution model testing, and we doing this by employing IQ-tree, by performing a full tree search in IQ-tree, and in the process of that, also estimating the best fitting substitution models. And we also, so Velociraptor collects all kinds of statistics during this process, which can also then help us again to create different subsets of the data based on the average bootstrap support, for example, of the gene tree. So, and once we have the gene trees calculated and the best models calculated, we can perform the phylogenomic analysis. And we provide 3 different algorithmic approaches, a neighbor joining approach, a species tree approach with Astral, and 2 different maximum likelihood tree approaches with IQ-tree and RAXML and G. And once this is done, you end up with a bunch of different trees that correspond to all the different combinations of aligners and trimmers that you selected during your previous steps. So, but this is not the end of it. We have included many additional features, extensive customization of different parts and options, all kinds of ways to parallelize all of this. And we also, as I just said, we provide different results for different parameters and software combinations, but also for different random seeds if you want to go that deep and try that as well. Moreover, we have detailed reporting of intermediate results as HTML and PDF files, and we have some comprehensive functionality to plot and compare tree topologies. So what you would get from some of these additional features and reporting functionalities is what you see here. So this is an example analysis done with Velociraptor, and the figures here are exactly as they have been spit out by Velociraptor. So the figures that you get are basically almost publication ready. So the only thing I did is I arranged them into this panel here. So what are we looking at? We're looking at, for example, a summary of the statistics that have been collected during the different steps. So genome download, for example, how many genomes have been successfully downloaded and so on. Or for example, with model test, we get pie charts telling us how many, what the different models were that have been estimated during this model testing step. When the whole analysis is finished, we can calculate a PCA based on tip-to-tip distances of these large-scale phylogenomic trees and plot them. And then we can easily spot differences, methodological differences or differences that large-scale rearrangements, for example, in the topology. We can then go deeper and compare individual trees if we want, based on quartet subtrees, and all the tree plotting and labeling here with the taxonomic information is done automatically by querying the NCBI databases, and you can select the taxonomic level that you want to annotate, for example, on the tree. And in this case, we compare the only difference, or basically there are two differences here. So this tree was calculated with IQ-tree, and this one with RAXML. And this one was aligned with MAFT, and this one was aligned with muscle. We can also look at something similar here as well, looking at all trees. We get this similarity heat map of all trees based on quartet subtrees. Oh, and yeah, I forgot to mention that the conflict that you get between these two topologies will be highlighted in red here in these tree plots. So, but this was just a very small example. So we ran Velociraptor also on 3 large-scale data sets based on data downloaded from NCBI. And these data sets come from arthropods, from vertebrates, and from fungi. And there's not a lot of time left, but I will quickly show you those trees. So this is the arthropod tree. You have the Velociraptor settings that were used here. And this would be the fungal tree. This is a very large tree in terms of the number of genomes. It's like 2,300 genomes almost that were used for this. But in terms of alignment length, the vertebrate tree beat the 2 other examples with over 600,000 amino acid sites that have been used here. And I don't have a fancy plot, unfortunately, for that one now, but we printed it out on paper and pinned it to the wall at our institute. In case you are wondering now where you are, that would be here. So that would be the mammal clade. And yeah, so with that, I'm already at the end of my talk. I hope I could get you interested in our small piece of software. You can scan the QR code or go to my GitHub page to try it out. We have a small test data set with 10 genomes that runs on a normal laptop in about an hour. So you can test it yourself if you want without having large computational power available. So with that, thank you for your attention, and I'm happy to take questions if there is time for that.
Excellent talk. I think we can take a question while we do our changeover. So if anybody does have a question for our presenter, you can put up your hand or put the question in the chat. And since we are a little tight on time, Charlie, you can go ahead and get your slide share up while we are taking questions.
Sure thing.
Not so much a question as a comment. I really liked how you had the conflict on the trees highlighted and sort of doing the PCA among the tips of 2 estimated trees. I think is neat, because one of the big problems I think with a lot of the standardization and the reproducibility is sort of the lack of good visualizations for uncertainty. So
Yeah.
really
I
cool
think so.
important.
I think so. True, and that's true. And I think that you know, when you run a full file disruptor analysis, then you end up with many trees again. So it's not that you get one solution and you need to somehow make sense of this whole forest of trees, you know. And and I think that's why we tried to come up with something that is intuitive and yeah, visually appealing also. But thank you. That's very well appreciated.
All right, thank you. And Charlie, you are welcome to start whenever you are ready.
All right, can you hear me?
Loud and clear.
Okay. All right, thank you. Yeah, so my name is Charlie. I'm a postdoc working in Dalhousie University in Canada. And today I'm going to be showing you some of the work that I'm carrying out as part of the Andrew Roger and Ed Susko labs into developing site and branch heterogeneous models of protein evolution. Okay, so why would you want to model site and branch heterogeneity in the first place? Well, the phylogenetic data sets that we use to make trees generally tend to incorporate very different proteins from species that have very different lifestyle or environmental adaptations. And so you're sticking all of these proteins together, and if you don't have a model which is able to account for the varying evolutionary processes that occur at the site and branch level of your data set, you may induce long branch attraction artifacts, and it'll give you an artifactual tree. So I have an example here at the bottom that I'll discuss in more detail a bit later in the talk. So in terms of modeling site heterogeneity, this is readily achievable using mixture models that are implemented in standard tools like IQ-tree or Phylobase. Usually we talk about profile mixture models. So these are models which assume that the individual site-specific processes in a data set can be adequately described using a set distribution of site process profiles. So these may be mixtures that are based on empirical data, such as the C-series of models, C10 to C60, or they may be larger sets that are generated using a Markov chain approach, as in the more complex CAT model that you'd find in Phylobase. Now, for modeling branch heterogeneity, there are also models that can account for branch heterogeneity, and they do a pretty good job of it. But their use comes with some caveats, and the basic caveat is that modeling branch heterogeneity with these models tends to be extremely slow and requires a lot of computational resources that are usually infeasible for a lot of projects. Other caveats include, for example, in the NDCH model, that this can't be combined with site heterogeneous approaches. So you're sacrificing the ability to model site heterogeneity for the ability to model branch heterogeneity. So previous work carried out in the Roger and Susko labs, led by Sergio Muñoz Gomez, developed a new model called GFMix, which is able to account for site and branch heterogeneity. And Sergio and company applied it to try and resolve the origin of mitochondria. So it's been difficult to place exactly where mitochondria appear in trees in relation to alpha proteobacteria, largely because there's a high degree of compositional variation of GARP and thymic amino acids within the alpha proteobacteria due to certain phyla within alpha proteobacteria having very different GC versus AT content in their genomes. So to give a brief summary of what they were able to do, once they applied this GFMix model to their data set, they were able to recover a alpha proteobacteria sister tree where the mitochondria are basically sister to all alpha proteobacteria without having to do any additional treatment of the data set, like removing compositional outliers or recoding the amino acids into biochemical groups. So you're able to improve the model rather than having to do anything with the data. So what GFMix does, to give a brief summary, is it works on top of an existing mixture model and introduces what's called a new B parameter. So for every branch in a given tree, BE represents the ratio of GARP to thymic amino acids at that branch. If your branch is a tip, you calculate BE directly from sequence data. If it's an internal branch, BE is estimated from all branches descendant from that branch E. So these BEs are used to modify the mixture model at each branch so that the frequencies of each profile in the model are modified to reflect the BE ratio. This is done using linear equations. And then you can re-estimate the likelihood of the tree with your new model. So what I'm going to be talking about for the rest of this talk is my own work, which is developing and extending this GF mix model and then looking to apply it to some data sets that we know are subject to compositional heterogeneity. So I'll take each of these bullet points individually. So first off, there's the GF mix software itself. So this is written by Professor Ed Susko at Dalhousie. It's primarily written in R with the likelihood estimation part written in C. So what I've been doing has been writing a custom sort of R wrapper or package. I've been reading a lot of Hadley Wickham's stuff lately that overlays this GF mix software with some new features. The main feature being that we now allow GF mix to model any composition ratio to act as your B parameter. So we're not just restricting the potential amino acid composition bias to GARP and Phi-Mink. And then there's also some other quality of life improvements as well. And then we're also working on other improvements to the parameters of the model, such as how we exactly estimate B at each branch, but I won't have time to talk about that today. So what we want to do is we want to be able to take an input data set and figure out what's the most appropriate composition bias to actually model with GF mix. And so we've been trying out a number of different approaches for this, but what seems to be the best approach at the moment is to use a binomial test of two proportions. So the basic idea is you count up all 20 amino acids in your data set across all your species. And then you divide those species into two groups, either using some a priori knowledge you have, or you can use another statistical test to divide these species up. And so you have your counts for one part of the data set, your counts for another part of the data set, and for each amino acid, you feed those counts into that Z equation that I have there on the top left, which is the binomial test of two proportions. So this will give us a Z score for each amino acid. So we place amino acids with a Z score greater than 1.96 into our G class, amino acids with a score less than minus 1.96 into our F class, and all the other amino acids go into our other class. So G and F are basically the classes of amino acids that are significantly different between the two groups in your data set. So what we're also looking at trying to figure out is how good are our selection models at actually identifying the correct compositional heterogeneity when we already know what it's going to be. And so how we're doing that is using simulated data. So I'm using the AliSim software, which is part of IQ-Tree, which allows you to simulate alignment data under complex models of evolution. So the simulations I've been working with are just using a simple 4-taxon tree, taxa A, B, C, and D, where I have set the underlying GARP to Phi-Mink composition ratio for taxa A and C to 1, and taxa B and D to 0.2. So what that means is B and D will have a lot more Phi-Mink amino acids than GARP amino acids. This is quite an involved process, so I won't go into too much more detail as to how to set the simulations up, but what I can show you is that if you run the simulation 100 times and you generate 100 alignments that are simulated under GARP Phi-Mink conditions, I then ran the binomial approach to see how many times do we get GARP amino acids falling into our G group, Phi-Mink amino acids falling into our F group, and the other amino acids falling into the other class. And so on the plot here on the bottom, I've got percentage GARP, percentage Phi-Mink, percentage other. So across all of our replicates, GARP seems to be being identified within the G class. Phi-Mink is usually identified within the F class, but what's happening with the other amino acids is that some of them are going into the G class and some of them are going into the F class. So we have to look in a bit more detail as to what's going on there. But we are getting mostly GARP and mostly Phi-Mink in our G and F class. So For the last couple of slides, I'm going to talk about how we've been applying GFMix to already published data or empirical data. So to go back to my first slide, these are trees taken from a paper by Peter Foster and Donal Hickey from 1998, where they had a data set of 12 mitochondrial genes sampled from 8 taxa. So you've got your 2 animals, Allomyces as your fungus, and then 5 insects or bugs. So the tree on the left is the correct biological tree, which should follow the ecdysozoa hypothesis, where your nematodes place at the base of the ecdysozoa clade, right next to brine shrimp. However, when they tried making a tree from this 12 gene data set using approaches which don't account for branch heterogeneity, what ends up happening is that honeybee and nematode get stuck together in the tree. And this is because both those sequences have elevated numbers of thymine amino acids, even though we know that honeybee and nematode are not each other's closest relative, even within this tree. So showing a kind of a draft analysis here. So I carried out reconstruction of the tree using LG C60 plus gamma, and then another reconstruction using our GFMix model in addition to C60, LG, and gamma. And so unfortunately, we don't get the correct tree as being one of both topologies with the highest support. But what we do see is that the difference between the long branch attraction tree on the top and the correct tree on the bottom, the difference between those decreases from 15 likelihood units when you just use C60 to 5 likelihood units when you add GFMix in there. So we are reducing some of the artifactual support for the long branch attraction tree. And so I mentioned that we're looking at ways of new ways of improving some of the internal parameters of the model, such as the B-value estimation. So maybe once we apply those, we may see the correct tree being better supported than the long branch attraction tree. And so then the final example is related to placing, figuring out the root of eukaryotes. So Kelsey Williamson, who's a PhD student in Andrew Rogers' lab in Dalhousie, has built a 93 gene data set, which contains data from 63 eukaryotes and 37 alpha proteobacteria. And so she's rooting eukaryotes in relation to alpha proteobacteria using the outgroup rooting method. So much of her analysis with another postdoc in the lab, Hector Banos, using various different models, suggests that there is an opimoda-like root for the eukaryotes, which I think is shown there on the top right of that slide. However, it's unclear exactly what is going on with the anamoeba taxa. They're highlighted in blue there on the right-hand side of the slide. They seem to either place within this clade here at the top or at the base of the eukaryotes. And we think the reason that this is happening is because they have low site occupancy, and this is a very heterogeneous data set. So basically, this draft GFMix analysis here is consistent with other approaches in that it chooses an opimoda-like route for the eukaryotes. We see improved likelihoods when we move from using site only to site and branch heterogeneity, and there's slightly better support for the opimoda-like route over the metamonada route compared to other models. So yeah, I think that's about everything. So I'd like to thank everyone I work with at DAL. We have some overseas collaborators as well in Australia and France, and the funding bodies for funding this research. So thank you.
All right, wonderful talk. And we do have a little bit of time for questions. So if anybody does have a question, you are certainly welcome to. Perfect. Daniel, I think you should be able to unmute yourself still.
Yep.
Perfect.
Okay, cool. First off, great talk. It's something that I've kind of considered because coming off our last conversation with April, what I'm kind of coming from, nematodes tend to have long branch attraction, which is a pain in the butt to have to deal with. So I'm glad you really point that out. But I guess my question as a microscopist, and I promise I have a tie in here, with confocal, you can change your parameters so much that you start creating data. So at what point do you kind of call, I've manipulated these trees too much and now it's false, versus knowing when to cut that off? I don't know if that's like a very clear question, but that's the same kind of question that I've always asked, like, depending on what species do you include, what species do you not include, because sometimes those calls can change the shape of your tree, and sometimes maybe enough to even change your data. So how do you determine when to stop before you start creating data?
Those are all very good points. I think what I would say is that we're not creating data in the sense of we're adding additional sites in, or we're adding additional species in. I think what we're trying to do is we're trying to improve the model in such a way that it better reflects the data. Now, they're running the risk that you over parameterize the model. So you introduce too many parameters in because you're trying to account for too much stuff. I think there's other work from postdocs in our lab showing that, say the C60 example isn't overfit. I don't think this would be a problem with GF mix either, but that's definitely that's a valid concern, definitely.
Excellent. Well, we are